Maybe I can start with like, I think general challenges that we have is really understanding gene regulatory elements in connection to gene expression. And especially since I think both of the modalities, even in single modality assays, are sparse. So it's really challenging, especially for ataxic and chromatin accessibility, to identify the elements which are really capable to drive and affect gene expression in a quantitative manner. And so I think therefore combining chromatin accessibility and RNA-seq, for example, is a big step forward. However, we still have the problem that while now we both of the modalities in the same cell, it's still sparse and even sparser than we do the single modality. So it still becomes a little bit tricky to connect elements to their cognitive target genes.
Great. Other folks want to add to that? I mean, one of the issues I think related to understanding regulatory elements is to be able to get to a level where you can identify individual transcription factor binding sites in single cells, right? This is still a big challenge, right? You can get down to small numbers of cells, but you know, you can infer obviously what might be happening, but it would actually be useful to get that. I know labs are trying to get to that point, but I think adding to that would be the ability to actually visualize or capture that information in a single cell.
Another thing that I could think also like what's currently in infancy is like profiling histone modifications in single cells, which can kind of add an additional layer of complexity to chromatin accessibility and DNA methylome single cell data in terms of like really identifying what might be active elements, what might be insulator elements, etc., what might be structural elements.
Yeah, I think with some of the advances with like cut and tag and those types of assays, it kind of opens the door to targeting different histone modifications through some of the existing, you know, molecular tools that we have available. So, you know, using that potentially alongside RNA or other properties as well.
So what are the, maybe you mentioned a bit already, Sebastian, but what are the modalities that you would make most sense to have a co-assay for? You know, regulatory elements and transcription and other epigenetic marks, methylation, all three at the same time, protein assays, what other microRNAs from the same cell, what would make the most sense as for co-assay? Everything from every cell, I guess.
I just wanted to mention one thing that was very interesting that I heard at AGBT from 10X was that they're going to be partnering with companies to look at intracellular protein signaling events. I think that potentially could open up a lot that's not available right now.
So Josh, what specifically were they, you mean like changes in protein phosphorylation, or was it protein?
Right.
Yeah.
They partnered with cell signaling technologies and BioLegend, I think, and they didn't any specifics or show any data, but you can imagine if you think a particular pathway is activated or not activated, you could get a lot of information that way. I guess that's layered on top of RNA-seq.
Mm-hmm. Yeah, I think one of the common themes is a lot of these additional properties are layered on with RNA-seq. So, you know, in the document we have that, you know, RNA is kind of the bridge between a lot of these different properties. So, you know, just a lot of efforts to get, you know, RNA and something else where, you know, that kind of enables integration across all of these other properties, you know, using RNA as that bridge. So I think that's largely been the predominant kind of bridging property, but potentially other ones could be used. I think RNA is the most accessible, so that's largely why it's been used. But if there are thoughts on other properties, that'd be interesting too.
One thing that I'm wondering if it might make sense to have also like because I'm not aware that like proteome and like really whole cell proteome in combination with RNA-seq, because I think so far we always, as you say, we use RNA as a bridge, but what does it really mean? What ends up in the cell as a protein and with functional? So I don't know if there are molecular ways to actually measure RNA and protein from the same cell.
So there's definitely things like site-seq where you've got the cell surface proteins, and there have been some like RAID, I believe is the name of it, where it's similar with antibodies that bind to proteins in the nucleus even, with tags that you can read out with single-cell RNA-seq assays. So I think that's, there's not a whole lot of work that's been done on it that I've seen. I don't know if there are people in different consortia that have been deploying those types of strategies to look at not just cell surface markers, but other proteins in the cell and their relative abundances.
So maybe for folks, so we've talked about a bunch of methods so far. Is any consortium member here on this call doing something else that could be, is there any method that you haven't heard or that you guys are doing that wasn't mentioned? Yeah, I think I agree. I think that the kind of the beginnings of some of these other assays could be very, very useful, depending on the question, to measure the actual changes in protein abundance or phosphorylation in a certain circumstance.
So maybe I can mention about my challenge for the adipocyte field, because the size of the cells and also the cells is really easy to be very fragile. So we couldn't really do single cell RNA sequencing. We have to go with single nucleus RNA sequencing. So with that limitations, I think some of the core assay proteins, at this with the current technology, I don't know if anyone could help us. It doesn't seem to be able to apply in the cell type that we are interested. I just want to share this with the group. Yeah, okay, maybe so some other people has experience could help us.
So some other issues, that's a very good point, that were related to the fragility or the size. Right. And so some of the assays that require cytoplasmic or membrane protein detection can't be applied necessarily across. So maybe that's methods for enriching or extracting cells that don't fit into droplets or wells or et cetera. So methodologies to survey larger collections of cell types that are not kind of amenable to the standard protocol at this point, nonetheless important.
I want to add something here. I'm part of the aging consortium. Now that we were talking about nuclear RNA-seq, I think it's really important, and in particular because it's opening the chance for us to look in our freezers, liquid nitrogen tanks, and look big biobanks that we have there. We have collected for many, many years. Yeah, we may miss part of the proteins or the markers in the surface, but that will open the chance to really go for large numbers of samples. In addition, I always want to remind everybody about basic information that we need always to have in hand. Because, for example, we talk about methylation, but methylation is clearly changed with age. Now we know about, I learned about today about the effect of smoking. I mean, we need to go back and have a really good basic information about the samples that we're running. It's not a matter of go and run 100 samples and but we need to be very sure about the clinical information about those samples. I want to emphasize that. And that's been critical for our lone aging consortium. And like I say, it's not always easy to get fresh lung samples to run single cell. But with the nuclear, we can open many samples that we all have in the freezer.
Another thought that I had related to this, what I think is important to think about, like how many samples, like how many donors do we need to profile, and how many cells from each donor to get like a really reliable answer, kind of like a power analysis in that sense.
Jay here. I'd like to go back to the question of multi-modality. So at Beacon, we've been working a lot on 5' strategy, and although we are just focusing on the transcriptome, we're able to capture enhancer RNAs, and we see very strong correlation with open chromatin accessibility. Not 100%, but we're trying to distinguish what is the difference between enhancers that are open without the enhancer RNA expression. What are the enhancers with the enhancer transcription? Is there a functional distinction between open chromatin accessibility? And so these are the questions that we're able to address with 5' strategy in the single cell resolution. Hello? Yeah. Maybe another.
Yeah, somebody, can you mute yourself? There's some background noise. Who is it? Bao?
Yeah.
I'm seeing if I can find out who it is, Joe. Mute them when I can. I just want to check in and make sure everybody's okay. And
yeah, we're good.
You've got some scribes and capturing the data as you go.
Yes, we're good. We've got 2 scribes.
Excellent. I'll try and find whoever it is who's got the background noise. I don't mean to disturb. Off you go.
Okay, so is that assay the 5' assay? So, so adding essentially additional coverage information, full length, potentially 5', 3' to some of the existing assays where it's not readily accessible with for the different consortium members. So that's essentially the increasing the coverage, the quality also of the utility of that data in one assay. So for RNA-seq, adding potential 5' provides additional information that correlates with some of the other kinds of data and reinforces it.
Yeah, I think that brings up another good point too. In a lot of assays, I guess for lack of a better word, you get something that's like splash information into another modality, right? So, in that case with enhancer RNAs, you're getting information into, active, enhancers. So, even though you're not directly assaying that, you're getting kind of splash information there that can help you tie modalities together. And it also kind of leads to some questions around, what's kind of the optimum way to structure a co-assay? Are you going to get really high quality information of one property and then some, low, really sparse information on a second property versus try and balance them equally, versus, maybe lower quality for both or kind of how should that be structured? And I think it, definitely depends on the modality that you're looking at in terms of what you can get with what level of information. But I think it's an important question, and it'd be interesting to hear what people's thoughts are on that with respect to how the modalities are balanced.
Right, and the additional cost for adding that, I mean, for some cases it could be warranted, basically could come along for the ride. In other cases, it's gonna add some additional cost to that assay. So what's the sort of, what would be the low-hanging fruit to be able to add, as you said, the splash assay? I think that people, if maybe sort of best practices, if you're going to measure this, this would add value, more value than something else.
I think that idea of adding value, I think that's something that our consortium or our standards of working group is trying to do. We're going to try and collect data on the same sample with multiple assays, and then computationally see what additional value there is from each one. That's with Holger Hein. I think he's on this group someplace, but he's quiet.
Holger, if you're on, you can chime in if you want. I don't. Maybe he moved to a different room. I don't see.
Oh, okay.
Yeah, well, I mean, there's multi-omic assays from a same, but you raised a good point, Josh. You're talking about integrating data from using the same sample material, and what are the advantages of sort of computational integration versus trying to do a sort of multi-omic assay, where you could go deep, really deep, for example, on the individual assays that haven't been developed for sort of multi-omics. What are, I guess maybe other sessions might be covering this, but the ability to integrate the data, having some subset of data that's ground truth. What are people's thoughts that are more computational? Maybe Christoph, you guys doing a lot of data integration from single modality assays, and how well do you think that fits with the multimodal assays when you have some ground truth from those cells?
Yeah, quick comment on that one. I think we are really going to be in a situation where there is one or several very high throughput assays that people will always be doing, and a lot of inference, like we've also seen with the epigenomics and various other consortia, where you would encode also, where you have those complicated stuff measured in a few cell types where you can really do it because you have the numbers that you need and you have the standardization that you need. And then you would try to propagate that to a lot of other cell types through correlation. And I guess that is something that hasn't quite caught on that much in the human cell atlas community, but that's clearly something we should be doing going forward. And clearly, those things that Andrew was already mentioning, that sometimes you can infer one from the other through enhanced RNAs, etc., that could provide a kind of connection point where you might have a relatively direct way of inferring from one to the other. But there might also be more indirect ways where you basically capture cell states, and then you essentially have a function or that converts an RNA signature into a cell state signature that would then be converted into a methylation signature or a transcription factor or a DNA methylation or some other protein signatures based on deep learning models that you have trained in those cell types where you can actually afford doing many of those.
Great, thanks, Christoph. So maybe let's move on. We only have, I think, 15 more minutes. So Andrew, maybe you want to take the next one about sequencing and imaging.
Yeah, so you know, in terms of modalities and also tying into the other subtopic is, you know, how can we actually use single cell omics-based assays and tie it into imaging? I mean, there's tools for doing, you know, spatial-based strategies, but you know, thinking in terms of, you know, actual cell slurry, single cell approaches, you know, are there ways to capture modalities that could be most readily assessed in imaging assays? Particularly things like, you know, I'm thinking multiplex or like cyclic IF, things like that, where you're getting lots of different protein marks. What's the best way to capture modality at the single cell level with potentially other modalities to bridge the gap to those types of assays?
So are people doing, in different consortia, doing sort of a kind of around that theme, and doing things like that are on the call?
So in H10 we are, where we're taking, you know, the same biopsy and running a whole host of different assays, multiple sections for cyclic immunofluorescence, also single cell RNA-seq, single cell ATAC-seq, single cell genome, and trying to find the best way to integrate those. And You know, we're trying to keep as much spatial information as possible to use that as a way to bridge things, but you know, it's still fairly challenging to get, you know, a matrix of immunofluorescence signals and tying those back to different cells. So it'd be nice if there were, you know, other modalities that we could capture at the single cell RNA-seq level or single cell ATAC-seq level that could tie into that a little better. So it'd be interesting to hear what other people are doing.
Anybody?
I think we have something similar to that. We're not trying to tie the spatial in at the beginning. We're using the spatial at the end as sort of a verification that what the other assays have given us, giving that we think that spatial may be more of a truth set in the end.
Our group is working in multiplex. I mean, we can run up to 12 samples at once, and that it's been extremely helpful in reducing the cost and increasing the variability between experiments. That can be important.
So I'm just thinking, so what's the kind of QC that you can do in those fairly, you know, complicated scenarios? What do you use for your we have a question about sort of standards. Let's take that. Andrew, what do you guys do for validation of some of that data?
So with a lot of our samples, it's been pretty challenging. You know, often we get these biopsy samples that are very, very different from one another. So the QC standards are just kind of all over the place, particularly in cancer. I think it's very different in some other tissues where you can have a little more control over the tissue state. For instance, in brain, like, you know, postmortem time is you know, certainly a valuable metric. So it's been challenging. We haven't processed a large number of samples yet, and we don't intend to do a very large number of samples for this initial study just because we're doing so many different modalities from each small sample. So that's been a challenge too. I mean, the samples that perform really well on one assay will typically perform really well on all the assays. So it's still a little all over the place just with the variability of tumor samples.
Mm-hmm.
One thing that we have sometimes seen that sometimes modalities can depending on the tissue perform better or worse. Like, for example, we have seen instances where single nuclear RNA-seq worked very well on a sample type, and we took the very same sample and did single cell ATAC-seq, and it didn't work at all. It seems to be a trend. Even if you have it perfectly working for one condition, the other modality might not work. And I think this is that we can tackle when we do a single modality, but it might be a little bit more tricky when we think about multi-omic approaches where we try to test both from the same sample with the same nuclear prep.
Can you speculate what might be the reason? Because normally, like what we're seeing a lot is that on archival samples, RNA-seq doesn't work so well because the RNA has degraded, but the ATAC-seq, single-cell ATAC-seq works quite nicely, whereas kind of the other way around, it seems kind of less obvious why that would be.
Yeah, my interpretation of this is that for ATAC-seq you need to have intact chromatin, so it needs to be still wrapped around the DNA needs to be wrapped around the histone octamer. Whereas on the other hand, when you do single nucleus RNA-seq, you just need the single-stranded RNA molecule to be captured by the oligo-DT primer.
So the sample degradation issue is different for RNA than it is for the chromatin assay. In other words, you find cases where the RNA is quite stable, there's no degradation, but something about the condition has resulted in nucleosome dissociation or something. And so you get a very poor signal from exactly the same material.
So we've definitely seen similar things as well, certainly in brain. In tumor, I think the issue that we have is often samples will just be very necrotic and a lot of cell death where everything just kind of fails. But another note on that too, with the chromatin structure, some of our initial samples, the way the tissue was processed, it ended up being put on like this salt high salt substrate or something, which obviously high salt disrupts chromatin structure, and it would result in just the single cell attack-seq looking like single cell whole genome, where it was just very uniform random distribution of reads across the genome, whereas the RNA worked fairly well for that. But we see it the other way too, where often in our some of our brain tissue samples will get really good attack-seq, but the RNA is more degraded, so it can go either way. And I think if you're trying to do a co-assay to kind of bring it back to the core of this topic, the tissue preservation, finding ways to actually enable co-assays where you're going to preserve multiple modalities when you do the cell dissociation or tissue, however you're breaking up the tissue or preserving the tissue, is really going to be a critical component to move these efforts forward.
So sort of best practices for tissue sample preservation or collection for the different organs across this, I think would be, what are the best, for example, the cases that you've seen very high quality material for chromatin accessibility, how was that, what was the metadata around the sample collection? It would be good to, across the entire consortium, I'm sure there's a lot of expertise that could be combined to say, hey, do this, don't do this, right? And maybe that applies across organs, across tissues, that little tricks that people have found. We don't have a lot of time left, but one of the things that I think we could get to is the question of accessibility. A lot of the assays, what are the current challenges that users face to implement the multimodal assays? So, my experience for years ago with things, where it's very custom, is that it's not, it's not so easily to adopt, and when things get democratized, they get commercialized, maybe that's not for the experts, but the wider community then produces tons of data which go into, some organized database. And so these kinds of experiments would be very challenging to do in a high throughput. Nonetheless, they add to that. So what do you think about accessibility? I mean, when you approach companies that are trying to, they're like 10x, and tell them about another assay, they sort of look at you. I mean, they've got a commercial goal here, and now they're, maybe RNA and attack-seq is their next challenge. But what are your thoughts about accessibility and what the users, the bigger, wider community, could face? Should there be ways of, engaging, for example, commercial entities to across consortia to say, hey, we're leveraging all of these different NIH consortia. We need, you guys to implement this. It works pretty well, but it needs to be moved into a format that could be more universally utilized. That would add a huge value.
Yeah, I think a big component to that as well is kind of messaging to the broader scientific community beyond people that are, very, technology development focused, and, getting the message out of, what's the utility of these different, co-assays and things like that, because that kind of has to be there to make a company actually want to go into that space. So I think that's an important component as well.
See, Mike Pazin had a question here we could get to. It says, whole cell versus nuclear RNA are quite different. What is more common in the HCA hub map, and has anybody done any comparisons? Any thoughts on that? Josh, you guys have probably done a bunch of comparisons of whole cell versus nuclear RNA.
I haven't done it. I haven't published anything myself, but I've seen some nice stuff on kidney published from, I think, from Ben Humphreys, and there's probably also stuff from brain, from Allen Brain Atlas. And I think you begin to see differential, some cell types show up more when you dissociate, and others show up better with nuclei, because, for example, immune cells are definitely not going to show up as well with nuclei as they will with dissociation, because they're already separated. And so I think it's definitely going to give two different pictures. I think you're going to have to do both, or at least understand.
Matthias here. We just published a paper last week on JCI Insight where we reported the different comparison between single cell and single node coming out of the KPMP. And like Joshua mentioned, you do see different cellular population adequately represented in the different conditions. And obviously, processing artifacts are also differently represented in the different processings. So having a systematic comparison of these two technologies in place and to benchmark going forward, I think we will be very valuable. And defining which cell type and pathways are of your prime interest should guide you in the technologies to be utilized. What's clear is with single cell having ultimate careful attention to tissue processing details is key. And again, protocols by the AMP Lupus Nephritis Network have been specifically developed to procure in kidney tissue and rheumatoid arthritis samples, for example, tissue with an earmark towards inflammatory cells. Different protocols are in place to retain epithelial cell integrity. With single nuke, it appears that tissue processing in an OCT frozen material and cryosections are a good way to access even historic samples that have been stored for some period of time.
So what I'm thinking is that if you look in the nucleus, you're looking more at instantaneous transcription rates. And if you look in the cytoplasm, you're looking at steady state rates. So that was my question. Okay.
So now the real task, we've identified a number of issues, right? We have quite a long list of things that questions that I think are important questions to address with these multi-omic single-cell sequencing-based methods. Now we've got to come up with a list of recommendations that could allow this cross-consortium collaborations to continue. Maybe identify a set of recommendations that we would say that would be if these areas could be addressed in a cross-consortium way, that this would bring the whole field forward. And so from this long list, I think it says try to capture a minimum of 5 recommendations. We can have more that address the problems or the questions that we've chosen to take forward. So if you look at the list, I think everybody can kind of scroll through the list. What are the real important recommendations for this group that could lead to cross-consortia collaborations? What could we pull out, or how could we combine these? You know, I guess we should identify what the problem is. I try to define as clearly as we can what some of the problems are, and that how this group could try to solve these problems. And do we have the required expertise to be able to do that?
Yeah, I think one of the big problems that I see moving forward is going to be really figuring out tissue preservation type strategies that are going to be suitable to multiple properties. And I know, people have done that for individual modalities, but being able to actually have a preservation strategy that works on all of them for these types of coassays, particularly if you can archive tissues using that method, I think that would be particularly valuable in an area that the more information, the better across the variety of consortia will be important for that.
So, Andrew, do you think that step one in that would be to identify what folks have seen, for example, like the description that both you and Sebastian gave about sort of what are your experiences with that, and then maybe there's a set of recommendations that we could, or collaborations that we could try different preservation methods. I know in our collaboration with the Allen Institute and with the Leonardson Group, Sebastian, for example, has been trying a variety of methods for looking at nuclei preservation in shipping. and has learned quite a bit, I think, about, you know, what's possible, what you can, you know, what freezing conditions might be useful, et cetera. So maybe the groups that are doing these assays could come together to describe what the challenges have been and maybe what some of the solutions have been. And that would help kind of facilitate the cross-consortium collaboration related to this question.
Yeah, I think to add on this, I think one thing which is very important, that I don't think that there will be a one fits all tissues. I think it will be different from tissue type, just simply stratifying into muscle tissue, brain tissue, and, for example, epithelial tissues, that you need to treat them in a different way. And so I think it would be great, especially with this field-specific expertise and how to handle the tissue in a specific way. I think that would be super important to have.
Yeah, and I agree. I think a one-size-fits-all, unfortunately, is never going to be the case. I mean, we're even looking at, you know, on a sample to sample based on H&E staining, having different protocols to go down. So, you know, perhaps having some mechanism of sharing that information broadly to the different consortia would be valuable in terms of, you know, what's worked, what hasn't, and, you know, either like H&E or just a description of, you know, the properties of the tissue in that instance of how things worked or didn't, I think would be valuable. I know we're doing some of that in H10, but a lot of it is kind of all over the place. So, you know, I think consolidating that would be useful, but also, you know, spanning across the different consortia.
And I think one thing that it would also help to do is getting more comparable data sets. For example, if like within, I know that like the same tissue can be part of multiple consortia, but if the data are, or the samples are prepped in a widely different way, then it might not be easy, as we discussed before, about biases in terms of like how the tissue is handled, to compare the data across consortia. So I think we could come up with a kind of best practices how to deal with one tissue. Then it would be also like for the wider research community very beneficial.
I always want to bring up one thing which just seems tied into this, is that we need to have some QC metrics that we can use to assess these tissue preservation or other questions. So I think that might be another thing, is to think about some sort of cross-consortia acceptance of methods, QC metrics for the different methods. And
yeah, I think, you know, with respect to the actual modality readout, but also, you know, cell recovery, nuclear recovery, things like that as well, that'll probably be more across all the different modalities and tissues.
So what do we have two different questions there, or is that one related both to tissue quality and metrics to assess the outcome of the sequencing of that tissue?
I was thinking of it as a separate question.
Okay.
It's multiple efforts.
So one recommendation would be for tissue sample preservation strategies that they could be, that a group could coalesce around that question and provide information about what they've observed, and then maybe come up with a standard protocol for a tissue type for the best quality. And then also a separate question would be to develop QC metrics to be able to assess the quality of a tissue based on data from sequencing of that tissue by some particular assay.
Another thought too, I don't know if anyone's done a type of analysis where, you know, certain tissues, even if they're different organs, they might have similar properties, right? So, you know, actually abstracting from the tissue level, but actually focusing on the inherent like organization and structure and matrix, ECM of the different tissues, and how, if that actually translates, if you have two tissues that have similar ECM, but they, do they actually perform the same way in doing these preservation strategies and single cell strategies, or are there other inherent differences? So kind of potentially a meta-analysis of the different tissue types and categorizing them by kind of just a vector of different, inherent properties of the tissue. I don't know if someone's done that. I'd imagine something that's broad like the HCA might be doing that. But just thinking of how we've kind of worked on this with HTAN, with trying to categorize different individual tumors that can be very, have a wide span of variety, even from the same patient. So something like that, but spread to other tissues.
So is it both, Andrew, just capturing some additional information about the, like the metadata, like how the H&E stain looked, or, I mean, I'm just,
or other, just, the number of cell, like the cell density of the tissue, just, you can imagine a variety of properties. So, if someone wanted to come in and profile a tissue that really hasn't been extensively profiled before, you could maybe look up a set of properties and see, oh, well, based on the cellularity of my tissue, or the, properties of the ECM, how much, collagen is present, things like that, would give you a guidance in terms of what to implement as kind of the best idea based on these other properties, instead of just looking at individual tissues. Just an idea.
Yeah, no, I think capturing additional metadata about the sample that you might not be doing at this point, like you said, how much of, what is this? What is the characteristic look like? Like how fluorescent is it, for example, if you're doing, in spatial kinds of experiments, and how you would deal with that. Maybe some of those properties would carry across different, how you what tissue dissociation method, for example, works best, and you might start out there for each organ. How about other things in the list? We kind of go back and survey, what problems can we define that can be uniquely addressed by this group from this list? I guess the idea here is that we're trying to make recommendations for areas that could be cross-consortium collaboration, so that groups could focus on a question and address it by some experiments, that then the data could be shared for these very, generic kinds of issues that cross-cutting.
I think one thing that might be important to look at, we discussed this before, like a multi-omic approach that just gives 50% modalities. Is that better than having like on the same sample, but different kind of cells or nuclei used for each of the individual assays, and to have systematic analysis for a set of tissues to come to a conclusion what is better?
Right. So cross-consortium collaboration to determine whether individual modalities or multi-omic modalities, individual modalities that are computationally merged, is a better approach, or maybe can be compared to approaches of multi-omics, and what you gain from one or lose from the other. Because
I think also tying into that is, if you do a multi-omic approach, how to balance those two modalities, right? And that is another factor in terms of, if you're also going deep with individual modalities to integrate, if you're doing maybe a co-assay to bridge the gap between the two, how should that be balanced based on, these variety of properties, and figuring out what gives you the most utility in terms of data set integration.
Yeah, the groups that are doing that kind of integration of data of single modalities are often supposed to say, oh, we're going to be replaced by multi-omic methods. And then when we use their methods to test on the multimodal versus the sort of parallel approach, they actually work quite well. So I think if the groups that were doing the computational approaches and the actual experimentalists, or the ones that are maybe doing both, could get together and focus on this problem, then you could determine maybe which multimodal methods are easier to combine computationally, well, single mode methods to combine computationally, versus others that might have very different background challenges. So that could be an area of collaboration across the consortium, to have these sort of comparison of put data sets out there that could be used for these that are multimodal and single, from even potentially from the same group, where you've done all, the multimodal assay, the two single assays, and then do the comparison with different tools. I know that's been done to some extent, but across a variety of tissues, I think we could get at the question of what, which modalities, combinations, can be more useful maybe, or can be applied in certain cases versus others. So that would be something I think that you could work with across the consortia.
So it sounded like Andrew was doing something like that already, and it sounds like we might be doing something like that.
Yeah. Right? Andrew, have you done that? Are you doing a sort of head-to-head comparison?
To some degree. I mean, it's, sample variability is still the kind of kicker for that. So I mean, I think that'll persist as a challenge. But yeah, we've done some comparison, and we've tended to go more on the just like single modalities, maybe a little bit of co-assay, but often, one of the challenges too is getting that balance. But when you do the co-assay between the two modalities, and how rich is each one of those modalities. And that's varied, from often sample to sample, but sometimes, very much so tissue type to tissue type.
So we have sort of three areas for it now. One is sort of the tissue and preservation issue, the QC of such things, maybe a comparison of single versus multimodal methods and how they can be best integrated. How about from that list, what are the other recommendations for cross-consortium collaboration that could come out of these questions? Yeah.
I'm also seeing the chat now. I kind of had this to the side, so some people have commented there. Some of it was on the tissue dissociations and some great protocols there that are worth looking at. But most recently, the question of how many samples are needed for each single modality is noted that by Sanjay that's a question that does keep coming up, and fits into some of the previous topic, but also it could be a new one as well with how many cells. I think it's also a question of how deep you profile each cell with respect, and then how many cells you profile. You can kind of have a trade-off there. But then the next layer of information is also, if you're doing multiple modalities, kind of fitting all of that together. How many cells or samples would you need with multiple modalities? I think is an important question too.
Right. So these are sort of related to the throughput of some of these assays, and I think we had that as a cost versus throughput, deep versus wide. I think some of these things have been discussed in the past, but not necessarily for multimodal approaches.
Yeah. Yeah, and I saw in the notes, like power analyses, how does one perform a power analysis on a multimodal data set, and balancing the modalities and cell numbers and depth of information for each modality? I think that's going to be a challenge. It might feed into the analysis session in the afternoon. But I think it's an interesting question, certainly.
Yeah, I agree. Other thoughts about that? Number of samples that you've sort of thinking about with these single cell assays that can be quite difficult to carry out in a way relative to other assays, but yet have a lot more information. The cost per assay, what's the minimum number of I guess it depends on the question of the disease or the state that you're trying to analyze, but it is an important question because you typically, it's not hundreds or thousands of single cell experiments. It can be, but it's that's pretty expensive.
Bringing up that point of cost, one of the things that came up in the last session was some of these additional modalities are kind of freebies, right? So potentially kind of cataloging which ones are there that have a really low cost to add on, and would be useful to add that extra layer of information. So you're potentially identifying what sorts of modalities you can just add on another modality or within a couple steps of the protocol to get that additional information without too much of an increased cost. So they're kind of things that should really be pushed forward and made as widely available and accessible as possible because of the low cost burden to actually add those on.
So again, looking through the list, what are other areas that members of different consortia might try to take on a challenge to work together? Computational integration. I mean, what is the state of, I mean, I haven't visited the HCA sort of pages for every, standards. I know within code, we had, and then continue to add new standard operating procedures or QC for different kinds of data sets. Is there a way, is this something that, I know we mentioned this already for some QC of tissues, but what are best practices for, does each group have different QC standards for some of these multimodal assays in particular, to determine whether or not one of the two methods, as Sebastian was saying, works well, the other doesn't. What's the balance between those? When would you say it was a success to have a multimodal assay that gave you 2 or 3 different outputs, and all of them had to be above some minimum threshold? I don't know that there are such standards.
Yeah, I think there's been a lot of effort for data QC standards in the single modality space, but I think they definitely are going to have to be different for multimodal space. I mean, at least the methods that I've seen and tried in my group to date seem like there's there's lower quality typically when you're doing a co-assay for each individual modality than doing them alone. So figuring out what the sliding scale would be for that type of kind of QC standards, I think will be important.
So basically groups that are doing multimodal assays to try to come up with a consensus for the different multimodal assays that they're doing to say, well, what would be, an idea of a minimum standard for if you're doing, for example, ATAC-seq and RNA-seq from the same cell, as to when you would say that, how you would report that as pass some quality threshold for number of genes detected or number of reads per cell, et cetera. Because I just said I don't think those things exist right now for the multimodal assays.
Yeah.
Other things that we can think of here, what problems can we state that this group could potentially collaborate on that need to be challenges that haven't been addressed so far within, let's say, individual consortium, but might be best across.
I mean, I don't know if that's something kind of like to compare. Like, I know like a lot of people developing co-assays, but try to converge on like best practices, also like in how to generate this data, and on the computational side, maybe have a systematic comparison between outputs, for example, droplet-based, plate-based, combinatorial barcoding based approaches.
So can you restate that again, Sebastian? What was
basically to get a systematic comparison, like because the wet lab assays for multiomics, like for example chromatin and RNA, there are several methods now out there.
Yeah.
And people already generated data to actually try to compare these data sets and to come up with like an idea what might be like the best protocol, best practices, and recommendations across consortia.
So for groups that are doing multimodal assays with the same pairs or components to the assay, to do a comparison among the groups to identify, you know, what the best practices or what results are obtained from each method, whether it be a droplet or a combinatorial assay, presumably with a common tissue. Would you have to do that as a kind of experiment that to be done by saying, hey, we'll share this tissue and then you guys see what you can find and we'll see what we can find and come up with that?
I think it might be ideal maybe to start from the same tissue and then do the different assays on the very same tissue from the same source, and maybe have a unified pipeline that might show that there are different biases in the different approaches, but it might give an idea what might work better for one question or the other.
Yeah, it may depend on the tissue type as well as to what, yeah. You know, so you might have to do, you know, 3 tissues or decide on some subset of tissues that, because the assays may work differently based on the fragility of the cells or nuclei or et cetera, yeah. In one assay versus another assay. I mean, this would require presumably either some support. I mean, potentially groups have funding to do this, but at least if groups are working on the same tissue already, they could compare maybe even existing results, especially when the assays are in the early stage of development, whether it's worthwhile to continue, and sometimes it's questionable as to whether or not you want to continue with a method when something else appears and, you know, it looks great in the literature, but when you try to reduce it to practice, it might not work so well. I mean, there's a lot of things that could be troubleshoot, you know, why didn't this method work? If the groups that are on this call work together to find out, you know, what if one protocol could have some subtlety that, so essentially it would be to try to do a comparison between groups that are already doing multimodal assays to see whether or not we can identify protocols that work better in one tissue versus another.
Yeah, and we've seen some of that. You know, my lab kind of got out of the business of trying to make co-assays because they're quite challenging and a lot of dead ends. But we like to, you know, whenever we see a new paper come out, we try that strategy. But we also notice that some work well on some tissue types and some on others. So potentially to have something that's more unifying, if you want to have groups try it on the same tissue types, maybe, selecting a set of tissues that have a variety of these kind of like properties that I was talking about before, where you might have some that are really ECM dense, whereas some aren't, to try and kind of span that scope of the properties of these tissues, along with, just like cell line as a control.
Right. So maybe a recommendation, sort of a problem could be that, if you're developing a new method, here's a set of tissues that you should, potentially apply that to, maybe even in a bank, a GTEx or whatever that you can point to and say, hey, try your method on, on these tissues and a cell line, and then report it. And that'll be kind of a standard for everybody to do. And then you're comparing apples to apples. And people can follow that or not follow it. But, at least if there's some recommendation that is, if you're developing a new method, here's a set of diverse tissues, not a huge number, but at least pick from that. And then then there would be some overlap among the groups. And then you could actually go say, oh, okay, we'll get that tissue, we'll try it.
I like that idea. Tissue sample, if it's that same tissue type, like let's say your method works really well if you use this specific tissue preservation protocol, then you can just go ahead and do it on that. But a guideline of those tissue types I think would be valuable.
Yeah, I was just going to say that sounds like a good idea. I think if you just look at single cell RNA-seq, it looks like certain sample types have already become the de facto standards, and we have a preprint out for comparing the single cell RNA-seq method. There's many people who have done it, and a lot of people use PBMCs. A lot of people use a combination of a cell line and one from human, one from mouse. So we would be taking tissue types rather than those sample types. But if you standardize it, it'll make it easier. And I think also it allows benchmarking, because once one group does it, if another group has another assay, if you're the reviewer, you're going to say, well, why didn't you check it versus what this other group did on the same tissue type? So I think it will definitely help the field move forward quicker.
And I think it's important to include tissues, because also like from a tissue perspective, something like brain is fairly straightforward. And I think there are a lot of like nice comparable studies, and also for multi-omic analysis, there are several data sets out there, but especially for other tissue types, there's not so much known at the moment. So I think can be like 2 or 3 different tissue types that would be really nice.
Just sort of glancing through the chat, haven't really been looking at it as carefully as. Yeah, there was a question here. One that kind of comes to mind, but related to QC, is about the effects of dissociation of cell viability and gene expression. I know, for example, Chris Glass had a paper not so long ago showing, how microglia responds pretty much instantaneously, and their transcriptome profile is pretty dramatic. I'm not sure. So what the problem would have to. Maybe it's just reporting if for different tissues, what has been observed in terms of the response of the tissue to some dissociation protocol. Maybe as a cross consortium, it could be a forum about that. But if you isolate nuclei, what is one thing. But if you're trying to get whole cells out, I'm not sure what the problem is. I mean, I'm sure people have looked at it, but maybe it's a set of genes that rapidly change. If you're seeing this set of genes in this tissue, then there's a damage response, et cetera. Sort of like a list of that could be across tissue types that if you had a perfectly preserved situation where you were trying to minimally induce a response, these would be the genes that say that, hey, that you've activated some damage process. So sort of lifting, if people have done those experiments, to sort of make that on some database readily available, like you have blacklists for regions that look, bad in the genome that are very hard to map or have high, non-specificity. This could be a way to sort of put that in a common database.
Yeah, sort of just like, signal either as a set of genes or a set of loci that are indicative of, tissues or cells shifting their state as a response to the actual processing, and incorporate that as a QC metric by analyzing those specific signals.
Yeah, you're right. That would be part of the QC.
Another note too, something that we've come up with a lot in my group is cross-cell contamination with respect to either ambient chromatin or ambient RNA transcripts, and the huge difference between doing things like barn experiments on lines, which is what almost everything is on, versus primary tissues, and different modalities actually have different properties for that. So actually really encouraging people to do those types of barnyard experiments to quantify this ambient cross-cell contamination on their actual tissues, as opposed to cell lines in their the properties, because it is different with respect to different modalities. I think that's an important thing to think about.
So we restate that, a recommendation to actually carry out these sort of cross-contamination experiments on the actual tissues being, and not model tissues, not cell lines, for example.
Yeah, and I'm not specifically saying doublets. This came up in chat, doublet and multiplet removal. It's more the actual ambient. So, if you dissociate your cells, let's say a handful of cells, rupture, and their RNA is now either in solution, or maybe those transcripts are getting stuck to other cells. We definitely observe that. And same thing with things like ATAC, where you tagment and then some of the nuclei rupture, and you've got this ambient chromatin that could stick to other nuclei or get wedged in there, and actually quantifying that level. And I'm sure this is the same case with other modalities as well. So, actually doing that QC, and you don't need to do, like we just are now in the habit of spiking in, maybe 5% of our runs with a different organism in the same tissue from that organism. Or you can even use cell lines from another organism, just quantify the ambient modality from the sample you're looking at. So if you're doing human brain, we'll throw in 5% mouse cells. So
it would be a recommendation for maybe the same, if it for a protocol that would say, well, look, if everybody kind of did these experiments with a tissue of, at some ratio of that would be informative about how well that assay gave in terms of a cross-contamination of RNA or something. So maybe, how would we frame that would be, how would you work together to come up with a question that could be addressed across the consortium?
I think leveraging the diversity of the different tissues that are being profiled across the consortium is really where I think it'll be most valuable. If people from the variety of consortia run these types of assays on the primary tissue to look at cross-cell contamination, we could start to build a database of how much cross-cell contamination there is from just ambient material across the tissues, and potentially use those lessons in other consortia.
I could also think that it could feed into this list of genes that might be affected with artifacts related to cell dissociation. To come up with a set of lists, like, for example, for single nuclear RNA-seq, if there's one cell that very highly expressed one structural gene, then you see it all across the board. And it could be also like kind of like flag list of genes that you should check for in your data set. To maybe we could even kind of come up with a quantitative measure of like how good the preparation is based on some of these genes.
Okay, we have about a half an hour, is that right, to come up with for this final template, what problem we think we can solve by collaborating. It does, I guess, can be give your collaboration. So it can be more than one. I mean, I guess we could pick, you know, probably the most important thing that if this group or members of this group wanted to continue to work together on something, what would it be? You know, so we have a list of at least 5 recommendations or for cross collaborative, and this can be anybody, that can be other members, because there's a lot of people that could be in this group, but they're listening in the spatial, or they're going, you know, they're in another group, and but they're going to look at these recommendations. But for this particular group, and these final template will be posted, and also the ideas here. But what would be a problem that we think we can solve by collaborating, and what would be a recommendation on what we want to do? I mean, it could be, you know, lists of things, or it could be actual experiments. I said it could be these gene lists, or QC metrics that for multimodal assays, or what's the most important problem that we think we could address?
So I think, you know, part of it that really leverages the cross consortia is really the QC that are more designed to be in line with cross modality multimodal assays. I think that's, you know, very specific to this this subtopic, and you know, it's something that is distinct from, you know, some of the things in terms of cell tissue preservation can transcend just multimodal into single modality stuff, but really, you know, figuring out the QC metrics. And I did really like the idea of, you know, a common set of tissues that these assays should be tried on. So that's my thoughts on it.
Hey, Josh, do you want to maybe put that in the problems we think we can solve section in the blue box as one possibility? Or maybe it could be, restated, Andrew?
Yeah, so just developing a set of QC metrics that are specific to multimodal assays.
Okay, that's a very doable, I think, collaborative question that can be addressed. Okay, now recommendations, what we want to do. So specifically, there you go. UC standards of different, data standards for each single, okay. Yeah, so I think for the groups that are doing multimodal methods, this, and a place to put this information. I don't know if that's the kind of contribution of some of the folks at CZI. I think they have a portal to sort of discuss a lot of computational things. I'm not so familiar with that. Maybe folks on the call know more, but what would be the collaborative environment that you could discuss this? Is it Slack channel, or is it something else?
maybe like some sort of a document that people can just kind of list out what the metrics are. And yeah, I mean, Slack, it would be great too, and could be linked there, but some sort of so for H10, for instance, for each of the modalities across the consortia, we have like a document that lists what all the metadata should be, and a lot of that is QC metrics. Mm-hmm. So, and that seemed to work well. So something similar to that, but in multimodal focused and cross consortia.
Okay. Other ideas for areas that we could sort of copy paste that? I guess we have to fill it. What additional expertise do we need? Well, one would be that folks to create an environment to maybe have it in a sort of a database to post data, for example, a QC, like here's what was produced, here's an example of a set of data that, so you can actually look at it. Maybe use that as a kind of a benchmark for what you're comparing it to. Yeah, I think that the next question, so our next meeting is, so I don't know what the organizers of this have planned in terms of how they view the ways to, I mean, this group is kind of facilitates these kinds of things. I don't know if they have a plan for saying, hey, here's we need to give our group a collaborative title, but maybe they can facilitate this through this, the software that they're using for this platform. I don't know, this kind of storm. I mean, and then you would have maybe a once a month meeting or more frequently if it was thought that would be useful to address this sort of QC metrics for multimodal assays. So our next meeting is ? Depending on the ability to. I mean, this platform seems like it works pretty well. I mean, I guess because you have both the Zoom and the ability to do something, and it could be just Google Doc. It doesn't have to be their platform. How about, what was the other, how about this, I like the idea of a list of genes related to artifacts. Maybe that's part of this. So you have QC metrics, but you have, when things go off the rails, you have metrics that indicate that something is either contaminated or stressed tissue, something like that. I think that could be part of the QC, right?
Yeah, and possibly how that type of signature in one of the modalities could actually influence how you analyze the other. To bring the kind of connection between them, that could be useful.
I have a question about this. Isn't it possible that some of the same artifact activation by tissue preparation would also be important biological signature of wound healing or activated lymphocytes due to biology? So would part of this also be how to discriminate when this is an artifact and when this is a real biological signature?
Yeah, I think, the tissue dissociation, I guess you could use, for example, you could just snap freeze the tissue without, and then do bulk, and then deconvolution of the sample to try to figure out what no stress looks like. Otherwise, it's pretty hard to do it for, like in the brain, in an adult, you break all the processes off, you activate immediate early genes. Yeah, they're involved in neuronal signaling too, FOS and June, et cetera. But they'll also be activated. And so, yeah, so, you know, that's a good question, is the distinguishing between biology and experimental changes, normal biological changes and experimentally induced changes.
Yeah, and again, perhaps that's where, you know, multimodal assays could be particularly valuable, because if there's a certain modality that does change in response to stress more so than others, you know, you might be able to tease out some of those differences.
Yeah, that's a good point. Okay, so this looks like one area. Is there we could come up, is there something else we could come up with, a different problem that could be solved by collaborating that is not related to the QC metric? How about this question of number of samples and things for power analysis, you know, from existing data, where there's some biological signal that's known, you know, that you can see in a particular disease, for example, how many samples were required, depending on how much of the variance that can be explained by that, you know, change. I guess it's an issue. It'll change obviously for different diseases, how much signal you'll see, but I'm just wondering in general if there's a way to work on that for the folks that are like Christoph. Do you have any ideas about that?
Sorry, I'm doing stuff in parallel. What was the question?
Yeah, the question had to do with sample number. You know, is there a problem around that could be worked on across consortium about, you know, number of cells required per sample number, just not for that particular sample, but in general, the kind of calculations that are needed, so that people could have a guideline from, depending on how much signal they believe that they're looking for, how many samples would be required for single cell specifically, or multimodal assays. I'm just wondering about problems that could be, I think the experimental problems are a little harder because sending tissue around or things like that, but maybe another computational challenge beyond QC that could be used, that could be solved by these groups working together. And one issue has to do with, you know, numbers of cells, number of samples, the sensitivity of detection, things like that.
I think there's like at least 2 or 3 different approaches to this. The one that I think has worked well for other consortia where things were relatively clear is to define what is a reference map, basically. Like what was a reference genome in the International Human Epigenome Consortium. So to essentially put down hard criteria or semi-hard criteria and say this is what we would generally want. That I don't think has been done by the Human Cell Atlas, or has it been done in some areas of the Human Cell Atlas? Only within individual consortia, but not within, like we've clearly done it for our consortium, but I think across the Human Cell Atlas, there was not such a prescriptive statement, or was there? Or was there anything like gold, silver, and bronze Human Cell Atlas or data sets? Or what do you know what is there for RNA-seq?
Mm-hmm. Yeah, I don't know any sort of cross-consortium reference that you can use.
So if it hasn't happened yet for single-cell RNA-seq, then it's probably illusorical for multi-omics profiling, where things are kind of a lot more challenging anyway. Obviously, what can be done is to give some guidance on, like, not necessarily tell the people like you should have so many replicates, but give them some idea where we think that more replicates are needed and where less replicates are needed. For example, in our view, it was always that for our RNA-seq, we would do more replicates than for ChIP-seq, for example, just because we see more variability in the RNA-seq based on conditions in primary samples. And that perhaps that could be some way out to formulate best, or kind of what we consider qualitative good practices rather than quantitative criteria.
So these parameters haven't really been established for these multimodal assays for sure, right?
Like obviously what's going to be a big topic now with 10x launching their combined kits, whenever that's going to happen, where you can decide how deeply you sequence the RNA compartment versus how deeply you sequence the ataxic compartment. That will give rise to all kinds of approaches. Like in theory, that is a kit where everyone will be using more or less the same, but then some people will really throw 80% of their reads at the RNA-seq, other people will do the ataxic much deeper. And I think there, perhaps, it's a bit too early now because it seems 10x is not going to get that out anytime soon. But giving some advice, if you are doing multi-omics, should you go fulfill the minimum standards for each individual data type, or half of what you would normally do? And I think the first one where this question becomes real is for single-cell RNA-seq plus single-cell ataxic in the same assay.
Right. So so in preparation for that kind of data, or for existing assays like paired-seq or combinatorial single-cell combinatorial indexing that Andrews and Jay have worked on, that there will be data that where the comparison of samples may be challenging because of different read depths, different sensitivities, even though they're in the same assay. So you're thinking in terms of a gold standard for each modality, and then for the combined modalities. Is that?
Like, I guess a good, the combinatorial indexing is perhaps a good case here, because I guess, like Andrew would have to comment on that, but my guess is that what you get on combined RNA-seq and ataxic in a combinatorial indexing setup will always be orders of magnitude away from what you could get when you really do it in a plate-based, high-quality, low-throughput type of environment. So with, on the other hand, the throughput you get is so much higher that you wouldn't want to punish such type of assays for the low throughput, kind of low coverage, high scale. So I don't know, Andrew, what would you think is some, like, sticking for the moment with RNA-seq plus ataxic in the same assay, what could be some form of advice that would hold across the full spectrum of, on the one hand, combinatorial indexing, then 10x will probably be somewhere in between, and the plate-based, like Sarah Teichmann style assays where you can really invest a lot of effort on each individual cell, but with very limited throughput?
Yeah, I think, I mean, it's also with paired-seq, for instance, where you've got, potential to go really high throughput. And, I think all of these methods are evolving quite a bit. I know the stuff from the original SkyCar approach is pretty outdated data now, and there's always going to be improvements. So ideally, some of these really high throughput platforms will enable higher depth. I mean, we'll see what 10x comes out with in terms of, it's not just how much you sequence each, right, but how well your conversion of that underlying modality into sequenceable molecules was performed. And that, I think these various assays, certainly at the original combinatorial indexing approach that we were working on, getting the balance at the molecular biology side to actually capture both of those was challenging. And that's where I think most of the optimization has been. So yeah, I think how one would weight throughput versus actual depth per modality and balance between the modalities is going to be an important question. And I think it might vary quite a bit based on the sample that you're looking at and the questions that you're asking. So I think it's hard to put in place specific standards, with respect to that.
Well, one thing related to that, what I think is very important also to consider, especially for the ATAC-seq, not only sequencing depth, but also enrichment in the actual peak regions, because one thing that you might gain, for example, we see this in 10x data that actually you can get many more reads, but a lot of them are actually not in peak regions. So also like an idea of like, that ties in also back into the other kind of title, like what is the QC metrics that we have there? And I think it's always a trade-off between sequencing depth and sometimes also like signal to noise ratio for measurements.
Yeah, absolutely.
Hey, this is Will. I agree that QC measures are really important. I think one of the things that we could probably do is just choose a transcription start site set to be our standard for like TSS enrichments, which is a sort of a metric we really like to use to look for signal to noise. And because depending on what you use, you get variable answers. I think we have settled as a lab on one and happy to distribute that if that's interesting.
So this, thanks, Will. This sort of falls in line with the idea that defining references for these different modalities that people can standardize on and say, okay, here's what we see, here's what we see. I think that would be really valuable because we really don't have that kind of genomic view, gold, platinum, et cetera.
Yeah, I agree. I think having that set of TSS sites, as well as, when you're doing this enrichment, what windows you use, flanking it, right, is the background and defining that and the tool used. I would love to have someone come out with a universal tool that everyone starts using for that. So trying to get everyone on the same page, I think.
Hopefully we are. By the end of the week, I swear, we'll be posting something for integrated single cell attack, just attack, although it supports RNA attack integration, but yeah. So look, yeah, coming soon to a theater near you.
Well, I'll keep an eye out, certainly.
Okay, so do we have the right title for this, this other collaborative area of multimodal power analysis, or is this more of defining reference data, reference data modalities or reference data? I'm not sure. How do you want it? It's not just simply the number of cells. It's,
I mean, the TSS enrichment, I think, really falls into the QC.
Multimodal reference data. Yeah. We have a few more minutes left. How do we, okay, so for this topic, what additional expertise do we need? Well, we probably need to put this out there and get other groups that are that have been working on this problem across the consortia to just join in, because it'll be a little. I think it'll be these reference information will vary depending on the tissue type.
I guess in the last minutes, we can really just kind of focus on these two boxes, see if there's anything that needs to be added, just to try and wrap them up.
Yeah, I think so. One thing that's missing of these is, you know, we need an email contact, or at least one email contact. Other group members, so probably we would want to put more folks that would want to participate in these two areas, to just add. Certainly you could add my email to these, but I think probably folks that want to participate in these two, we can just add, maybe you can just add your own email to that.
Yeah, I'd definitely be interested in the QC, so I'll add mine for starters.
Folks want to add, can you, Sebastian, I think you were pretty eager about one of these. This QC would be useful to have you participate. I think essentially what we'll need to do is maybe capture folks all of the names of folks that have participated, and then when we do end up with identifying a time to maybe discuss this again, we can, and I'm sure other folks from the other breakout groups that want to participate in this can also sign up, but we need a mechanism to meet, and we'll have to rely on the organizers maybe to post these. This is, I think, this room at the end where you can kind of browse all of these questions, and then people can sign up based on their interest. So any other issues that for folks that maybe that have even been silent that want to join in the last 4 minutes, you can unmute yourselves and make comments and things that we've maybe missed that you'd like.
I have one question to kind of get the ball rolling. We've discussed in the first session about 5' versus 3' sequencing. Isn't that a form of multiomics in the first place? What happens, that someone must have done this, if you try to get 5' and 3', can you, or should you even try to get 5' and 3' sequencing in high throughput from the same cells? Would you gain anything there? Or is everyone shifting to 5', or everyone staying with 3', or people can't really quite decide? So what would be our recommendation there? Like if you do multiomics, I guess most of the multiomics protocols at the moment would combine 3' with whatever other modality, but perhaps except for the BCR TCR, obviously they use 5'. So what is our view there? Do we have any comment on whether multiomics profiling should go with 5' or with 3', or doesn't matter, or even combine both?
Yeah, I think that's a really good question. And I mean, it comes to also what we discussed earlier with what's the best bridging property, right? And it seems like RNA is the one that everyone's really settled on, but there's a lot of space there, especially with respect to 3' versus 5'.
Could this be part of the first kind of title, like the multimodal computation approaches for the power analysis, also to include something like this?
Yeah, I think that it could fit there. What's needed to bridge that.